So here I just want to illustrate a few so-called network motifs that are oftentimes seen in a lot of these networks. So a motif is basically just a type of recurring structure that tells you a little bit more information about how genes are related besides, gene A regulates gene B in a direct edge connection. And so here red boxes represent DNA sequences and blue represents proteins. And dashed lines basically represent either direct or indirect paths containing like 0 or more nodes. And so the first motif I want to talk a little bit about is autoregulation. So autoregulation is basically exactly what it sounds like. It's when, for example, some kind of regulator, it doesn't actually necessarily have to be a regulator, it can also be some kind of signaling molecule, basically gets expressed, and then either directly by, for example, through a transcription factor binding the enhancer, promoter, repressor regions of the same transcription factor, or indirectly through a bunch of other nodes, a gene can actually auto-regulate its own gene expression patterns. In what's known as a feed-forward loop, in a feed-forward loop, basically you can have a, for example, a regulator like MCM1, basically bind to and regulate the expression of another factor, SWI4, and SWI4 can go on to regulate the gene CLB2, but CLB2 can also be regulated by MCM1 directly. And so basically MCM1 influences the expression of CLB2 both directly and indirectly through SWI4. And so the reasons for this will become apparent later, but basically this allows MCM1 to have a little bit more finer control over how it influences the expression of CLB2. And finally, there's what's known as the multi-input motif, where basically an individual transcription factor can regulate many downstream target genes, but more often than not, transcription factors don't oftentimes act in isolation, they'll act as groups. And so in this case, FHL1, RAP1, and YAP5 might collectively bind many or the same targets. And what this potentially indicates, for example, is that not only do they regulate the same downstream genes, so they might potentially act in the same pathway, but what could also happen in addition to that is that they might be acting cooperatively. They might need to see each other's binding sites in order for regulation to happen. And so delving into the role of autoregulation for a moment, shown at the top in parts A, B, and C are 3 different ways in which regulation can really happen. Either the gene Y can directly influence gene X's expression in what's known as a simple regulation edge. You can have negative autoregulation where X basically negatively influences the expression of itself. Or you can have what's called positive autoregulation where X increases its own expression level. And so whether or not a gene, for example, negatively autoregulates itself, positively autoregulates itself, or acts through simple regulation, changes the dynamics of how signals are essentially sent through gene regulation. And so in this figure here in part D, basically what the y-axis represents is the expression level of gene X here, but it's scaled. So it's scaled by basically the rate at which it eventually, the expression rate that it eventually achieves once it has been activated and then has eventually reached steady state. And so the line in blue basically represents what happens if you have a relationship between gene Y and gene X, and gene Y just gets stimulated through some external stimulus and just starts increasing the expression level of X until it's steady state. And so if you think about how long it takes gene X to reach steady state kind of scale in terms of number of cell generations, then the blue line is essentially what you get. That's our baseline in this diagram. And so if you think about negative autoregulation, where X kind of inhibits its own expression. What's kind of, in some sense, counterintuitive, but is interesting is that what this typically leads to is a faster response in terms of the expression level of X basically achieves its steady state level of expression typically faster than it would under simple regulation. And part of the reason for this is that in negative autoregulation, the promoter of X tends to be strong. And so what that means is that X quickly binds, it tends to quickly bind to itself, for example. And also, the other reason for this is that when a gene negatively autoregulates itself, the steady state level of that gene is lower, because as soon as you turn on that gene, at some point it starts auto-regulating itself and turning its expression down. And so it reaches steady state faster. And so essentially negative autoregulation means that gene X tends to peak and tends to peak at its kind of steady state very fast. Whereas in contrast with positive autoregulation, the promoters, the binding site of, say, X on itself tends to be a little bit weaker. And also the steady state level of the gene is a little bit higher because the gene is like positively feedbacking on itself. It's able to turn itself up higher and higher and higher, typically. And so what that means is that it can take longer for X to kind of hit its maximum or its steady state level. And so this curve basically indicates that the response of X is essentially slower compared to simple regulation. And so just touching on feed-forward loops, at least one type of feed-forward loop. Suppose that you have a case where, again, X directly regulates gene Z, and X also regulates gene Z through Y, right? And suppose XX here stands for some stimulus, initial stimulus, which turns on X or can sustain expression of X. So consider in the top diagram, what would happen if, for example, that was what the input to X looked like. So basically X gets turned on twice, once with a short duration and one with a longer duration. If you look at the expression level of Y, basically the expression level of Y, it starts to become expressed as soon as X turns on, but for the short duration, because X never turns on for that long, Y never really is turned on for that long. And so it eventually decays quickly. And then when X is turned on for a longer period of time, then Y obviously starts to ramp up in expression before it starts decaying after X, the stimulus of X is not applied anymore. And so if you think about the expression level of Z, so what I drew in the diagram on the left is what's known as an AND gate. And what an AND gate means is that gene Z only turns on when both X and Y are bound, for example, to the promoter of Z in a high enough concentration. And so what you see is that when X is initially expressed at a very short interval, basically the dashed line on the graph in Y indicates kind of like the threshold at which Y is at a high enough concentration such that both X and Y are bound to the AND gate and therefore able to activate Z. And so the point here is that gene Z doesn't turn on at all when X is just turned on for a short duration. And even when X is turned on for a long duration, there's a delay between when Z starts being expressed and when Y is expressed, because Y has to ramp up to a high enough concentration before both X and Y are present and able to turn on Z. And so this, what's known as a sign-sensitive delay, means that having this AND gate there and requiring both X and Y to be present in order to turn on Z allows the cell to implement essentially a delay in expression of Z relative to X and Y. And so this is one way in which feed-forward loops can influence and change the expression level of target genes like Z through feed-forward loops instead of direct interactions. And there's other types of logic gates that you can use beside AND gates. You can use things like OR gates, which mean either X or Y present causes Z to be expressed, and so on. So one of the recurring features that people seem to see in a lot of different types of networks, including PPI networks, genetic interaction regulatory and co-expression networks is that a lot of these networks seem to be scale-free. And so what it intuitively means to be a scale-free network is that most nodes in your network will have very low degree. And so in this diagram on the right, for example, in this regulatory network, most promoters only have 1 or 2 regulators bound to it. And so what that also means is that there are a select few, relatively few nodes that have very high degree. And so those nodes that have very high degree are what are called hubs of a network. And so the interesting thing about hubs in a network is that there's generally been observed a strong correlation between how hubby or how like high of a degree a node has in a graph and how essential those genes are to the cell. And that kind of makes sense, right? Because if a gene is interacting with many other genes, then it's probably doing something important. And so if you like knock it out, for example, then it's probably lethal. And so again, it's kind of been interesting that this idea of scale-free networks is pretty prevalent, and it turns out that most of the networks that we talk about here, protein-protein, genetic interaction, regulatory, all of them generally seem to exhibit scale-free behaviors, regardless of which organism or cell type you're looking at. And so again, why is it important that these networks are scale-free? It tells you that, basically that tells you that there are hubs in the network. So there are nodes with extremely high degree, and those hubs tend to be really important genes. And so that's one way in which a network structure can tell you something about genes, because if the hubs in the network tend to be the really important ones, and those hubs can change depending on what type of cell or organism you're looking at. One interesting aspect about scale-free networks is that it actually makes your network, structurally wise anyways, robust to perturbation. So what I mean by that is that if most of your nodes in your network all have very low degree, only like 1 or 2 edges, then even if you knock out that gene, or if you knock it out or knock it down, in terms of the overall structure of the network, you're not really perturbing the global structure of the network that much. I think the only time you'll really perturb the structure of the network in a scale-free graph is if you start knocking out like hubs in the network, because those talk to a lot of other genes in the network. And so generally speaking, a lot of these scale-free networks are robust because if you don't hit a hub, then structurally wise, you haven't really changed that much. And basically, another way of saying the network is robust is that if you remove most nodes individually, then the overall connectivity of the network is still maintained. And yeah, so it's also worth pointing out that other related terms that are used to describe scale-free networks are also terms like power law or preferential detachment. Those mean pretty similar things as scale-free networks.